Good evening. Thank you all for joining us to our biology department speaker series. This semester, we are focusing on environment and biodiversity, and we are excited to have, especially
Good evening.
in March.
Thank you all for joining us to our biology department speaker series. This semester, we are focusing on environment.
Sorry about that. So we are excited to have in this month of Women in Science, Dr. Catherine Perez. Dr. Perez grew up living all over Texas. She attended Angelo State University for her bachelor's degree and master's degree, and the University of Alabama for her PhD, where she was an NSF IGRT Fellow in interdisciplinary freshwater sciences. She followed this with an NIH IRCDA postdoctoral training fellowship, which combined pedagogical training at the University of North Carolina, teaching at North Carolina Central, and a research laboratory at Duke University. She was then an assistant and associate professor at the University of Wisconsin, La Crosse, before moving to the University of Texas, Rio Grande Valley. Dr. Perez has two research areas: the evolution and ecology of snails and learning of evolution. In the snail area, her work has mostly focused on terrestrial and freshwater snails, addressing problems of invasive species and species of conservation concern. And currently, most of her research effort is going towards surveys and DNA work for cave and spring snails. Thank you for joining us, Dr. Perez. You have the floor.
Thank you. I appreciate the introduction. Okay, well, thank you everyone for joining me tonight. We'll try and keep the energy up. For me, it's at the end of a long day, so hopefully you guys are all here excited about learning some biology and maybe picking up a few interesting things to learn about snails. So as she introduced, my name is Catherine Perez, and I'm in the biology department at UTRGV in the Edinburgh campus. And I'm going to tell you today about the work of the students and collaborators and myself that all work in my lab on snails, mostly recently cave and spring snails that live in fresh water. Okay, so let me start off by telling you some of our goals. Broadly, very broadly speaking, we want to know more about Texas biodiversity, particularly the invertebrate biodiversity. This is important. We provide the foundational evidence that's needed to effectively conserve Texas biodiversity. And we do that by filling gaps in our knowledge and understanding of snail diversity and distributions. And bigger picture, I like to address some evolutionary questions about the drivers of morphological evolution and also speciation. One of the things that I really want to do, though, is have this conversation less with my friends. So I get text messages like this all the time. So this is from a colleague in San Marcos, Pete Diaz. He says, "Hey, Catherine, we got these from Comal Springs. Have you seen them before? They have some weird stripes on them, this interesting sculpture, these longitudinal lines." And so I had to say, no, I haven't seen these. And this is a little embarrassing, not me personally, but the fact is that Comal Springs is one of the best sampled springs in Texas. This is the spring that is the foundation for the Comal River and is in Landa Park in New Braunfels. It's a big spring and a big park where sort of everybody's family goes to picnic and for, like, you know, your little baby cousin's birthday party, you meet up at Landa Park. So everybody in the region goes there. It's really well sampled. And they're constantly pulling animals out of that spring that we've never seen before. But a few months later, I get the following message from one of my colleagues, a different one at Texas State in San Marcos, like, hey, I found a few of these bumpy snails from Barton Springs. What are they? They're really small. What are your thoughts? And of course, again, I have to say from Barton Springs, which is downtown Austin, where everybody in Austin goes to play in the spring, no idea what this snail is. And I want to have that conversation less often. I want to understand what species are out there, particularly those nearby in our backyard. Okay, so what I'm going to talk about today is some of what we've been working on cave snails, snails that live in the underground aquifers and caves in Central Texas. So I'll spend the first part of the talk about those cave snails and introduce some of the methods that we use. And then I'll tell you about my work on DNA barcoding and surveys in West Texas. Okay, so to just introduce this cave snail project. Here on the right is a picture from the top and the side of a cave snail. This is the crowned cave snail from some springs in Del Rio. And this is it positioned next to a grain of rice. So you can see the usual size of these animals. They're very, very small, usually maximum about 1 millimeter in size. So they are tiny, depigmented, eyeless, and all of that tininess makes them hard to work on. I've got a list here of some past and present students at UTRGV and a whole list of collaborators that work on this project with me. Okay, so some of the objectives of this diversity project are, like I said, to learn more about Texas biodiversity, to document snail diversity and distributions in groundwater systems in Central Texas, and then to identify and describe any new species that we find. And some of the reasons why this data is useful, it's, you know, of course, I can understand wondering, like, they're tiny snails that no one can see, so who cares? Some of the reasons why this data is useful and why various agencies fund our work is the U.S. Fish and Wildlife Service is charged with conserving the nation's organisms. And so this sort of data, where things occur and how the status of their populations, that aids in things like species status assessments, which help them determine which species should be considered threatened or endangered, and allows them to prioritize conservation funding. And then our recent work has been funded by the U.S. Army Corps of Engineers, who are interested in developing climate models for the groundwater in Central Texas. You probably understand that that stretch of Central Texas includes Del Rio, San Antonio, Austin, a whole swath of millions of people from the state who depend on that groundwater for their drinking water. And so the U.S. Army Corps wants to understand the resiliency of groundwater to climate change and other environmental perturbations. And just what organisms occur in the water is part of the information they need to build out those models to understand groundwater quality. Okay, so it introduced you to the snails that we know of in this system and show you where the aquifer is. This map on the right shows you the extent of the Edwards Trinity Aquifer system in Texas. So as I said, this broad swath of Central Texas, particularly I have focused a lot of my sampling between San Antonio and Austin. There are 14 currently described species. As you see, they have all different shapes. I'll talk a little bit more later about the ones that are tall. There's some that are flat, some that are a little bit more what we would call globose. They're a little bit taller, but not really tall. You have some that have ribs, some that are uncoiled partially, and then some that are uncoiled completely, and they're almost like a straight trumpet. So we have a really cool diversity of different morphological shapes of the shells. Just so you can get an idea, I'm going to talk in a moment about where these guys are specifically, but so you can have an idea of what they might be doing, what their role is in their ecosystem. As I said, they're tiny snails, but they can reach big abundances apparently where they have populations. We know they're primary consumers, so they are grazing on bacterial mats in the caves and aquifers. Sometimes those bacterial mats are formed using carbon from like leaves and things that wash into caves. In some places, in some parts of the aquifer, they're actually grazing on bacterial mats where the carbon source, the production is from, instead of photosynthesis, chemosynthesis. They're using some of the rocks and minerals in the water in order to make their own sugars. So they have a couple different sources of nutrients. These guys are eating bacterial mats. We know that the Texas blind salamanders in the genus Eurecia eat them because sometimes they get Eurecia, the little salamanders, and their guts will be full of snails. Some other potential predators are basically any secondary consumers, maybe the Texas blind catfish, isopods, amphipods, little crustaceans, and some flatworms. Now, as I said, these are in caves underground. We know pretty little about them specifically, but inferring from some of their relatives, we assume they probably live about a year, but we know things in cave systems live longer than things that are not, so that might be a little off. We're assuming about a year. We expect they have pretty narrow physiological ranges, that they're sensitive to changes in temperature and DO and environmental pollutants, but they have never been tested on any of these snails. We do know that they are egg layers, and so they're oviparous and lay egg capsules on hard rock or hard substrates that they find like each other. Okay, so now I want you to think about where exactly these little snails would be found underground, and think about this whole ecosystem. So you have bacteria lining a cave wall, like you see here in this cave pool, and then snails crawling around and eating that bacteria. And then things like little shrimps crawling around and eating the snails, and salamanders eating both. And I guess the top predator would be the salamander, and the catfish may be in that ecosystem. So they might be in a cave pool like this. More of them are probably going to be not in broad open cave settings, but in things like closed pools and the channels in between pools in an underground aquifer. So this shows like a cutaway view of a karst limestone aquifer system. And there's a bunch of places where water goes where these snails might be found. They might be found in cave pools, like you could see, but then if you look at all of these little channels, all of these channels that go between pools, they'll also be in those channels. And they might actually be more in those where there's a lot of surface area for bacteria to be growing. And then importantly, they're also going to be in the water that connects between the surface water and the groundwater. And so that's called the hyporheic zone. And so they'll be in the gravel connections between the river water and the groundwater below. And sometimes we can find them there. Imagine a snail that's way down here is going to be really hard for us to go get a sample of or study. And so we have to come up with other ways to find them. So here's a picture of a conduit, a big conduit that a diver could fit into where they might occur. More likely, they're going to be in this kind of Swiss cheese rock where, let's say, each of these holes is maybe the size of your fingertip, and they move through those channels. Okay, so I mentioned that it's hard to get to these places underground. One way that we can access the underground aquifer water is places where someone has dug a well or drilled a well. We can use the water coming up out of a well, filter that water and catch all the snails and other animals that come out, and make use of wells that people have already put. So here's an example of a well at the Uvalde Fish Hatchery where we have a net and a trap attached to the wellhead, and we filter all the water that comes through with a very fine mesh net, and then process and pick all of the snails and other animals out of that sample. Now, another place where we can take samples, instead of where humans have drilled a well down to the water, we can look for places where the water comes out of the aquifer naturally at things like springs. And so here is an example of where water is coming out from under a boulder, coming from the ground, from the aquifer at a spring. And we put a net so that it catches much of the flow of the spring, and the water flows through the net, and the little animals get trapped and caught up in what's called a bottle trap. Now, another way that we can access the aquifer is not in a well that's flowing, but in a well that someone has drilled and left open. We can put little traps called bottle traps down into the well and bait them with a little bit of pistachio, throw them in there, let that colonize, and then pull all the animals out. And then finally, I'll talk about this method a couple of times. This is called Bowrausch sampling. It's a way of sampling that water that connects between surface water and groundwater. So here we have driven a spike into the gravel next to a stream, and we're pumping water out that's connected to that stream water and to the groundwater. And we run that water through a filter or through a net to filter it, and then we keep that sample. And there's a whole ecosystem that just exists in that hyporheic zone. And so some of these snails we can catch in the hyporheic zone, which is the poking the pipe in and taking and pumping out a sample sample is a lot of work, but it's more possible than drilling new wells. Okay, and now my favorite sampling method would be going to a cool cave site and going down, going swimming, and looking for snails. One thing I want to remind you about, this is the view up. Here's the view down at the end of the ladder. One thing I want to remind you about is that these snails are tiny. So when I'm taking my samples in a cave, I don't necessarily know if I have found anything. So here's some bacteria mat lining some of the structures in the cave hanging down in the water. We don't actually know if we found anything. So often we take samples, spend all day or a week taking these samples, bring them back to the lab. We don't know until weeks later, until we process the sample, if we actually found our organisms. So we do the best we can and sample where they probably should be in the best ways to sample for them, and then bring our samples back. And so after you take your sample in the cave, process it a little bit in the field and preserve it, you get your reward. And at this particular spot, the whole family came. And so everybody got to have a nice time swimming and sort of reward the people who kept the small children from falling into the sinkholes while I took my samples. So that's my favorite sampling method. Okay, so what we found so far, this is a paper published by one of my former students, Dominique Alviar. What we found so far was we sampled 180 sites, wells, caves, and springs. And we, I think, about tripled the number of known sites where these snails are known from. So that was useful. We understand their distribution much better with this additional sampling. But what we were also trying to do was collect fresh snails with their tissues in there, so we could use that to look at their DNA. And so we have obtained tissue samples for all of the known species of snail and several that are unknown. So I just want to mention one example that I'll come back to in a moment when I show you some of the data from this. I showed you that there are tall cave snails, flat cave snails, and uncoiled cave snails. Here's the known or described species of tall cave snails from Central Texas. These two on the left are the ones that are known from the west, from Del Rio and from the Devil's River. So there's two that are described from there. And then this one on the right is known from Barton Springs. So this is Stygopergus bartonensis, the Barton Springs snail. And it has been proposed to be a candidate for federal listing, but the U.S. Fish and Wildlife Service could not list it because there wasn't enough known about it basically to list. And so we've been trying to find new locations where it's found and add in more information about the species. So we have, in addition to these known species of snail, these two from the west and one from the Austin area, we also have at least 4 types of snails that are tall that are unknown, including a smooth one from way up here in Bell County, this little guy that we just found a few months ago from the Big Bend area, this keeled one also from Del Rio, and then this one here that I'm going to talk about a little bit. This is the one that Pete sent me the text message about, saying, what is this thing from Comal Springs? Well, I really hoped that it was another population of Stygopergus bartonensis, because that would greatly expand the range, give us the idea that this species is much more threatened than we were afraid of. So I had tentatively said maybe it's Stygopergus bartonensis, just kind of a big range extension. But now we have to figure out what to do with these unknown animals. Okay, so in order to do that, we're going to build an evolutionary tree of the cave snails of Central Texas. And some of the questions that we want to ask are addressed with this evolutionary tree. Are all of the Texas cave snails close relatives? Or have multiple lineages adapted to cave conditions? So basically, how many times have this group of snails invaded cave environments in Texas? How does shape evolve in these cave snails, the flat versus the tall shapes? What species are there? How do we tell them apart? And are these unknown snails that we're not quite sure if they match any of the known species, are they new, or do they belong to existing species? And so to do that, we're going to use those tissues that we collected and build a DNA, an evolutionary tree based on their DNA. And so this is one way of drawing that evolutionary tree. I want to show you a couple others, because some evolutionary trees are going to be most of the data that you'll see in my talk. And so I want to make sure that everybody's on the same page with understanding how they work. So this is a drawing of an evolutionary tree. It's also called a phylogeny. This is a method to visualize the evolutionary history of an organism. So it's a way of drawing the relationships of organisms. So on this tree, you can see the two insects, the butterfly and the fly, are being drawn as being closely related to each other. They're connected by this short branch here, and that indicates they have a close relationship. So the two insects are shown to be most closely related. Then more distantly related to them is the snail, and the most distantly related to all of them is this bird. This underlying evolutionary tree is built from studying the characters that vary among these species. So for example, if we consider these four species, four groups of organisms, the butterfly and the fly share characters like having a compound eye, having two antennae, having 6 legs, 4 wings, exoskeletons of chitin, all sorts of insect features that they share in common that distinguish them from the snail and from the bird. And so those characteristics are used to support this idea that these are the most closely related. So a phylogeny can show you, or an evolutionary tree can show you which organisms are most closely related to each other. It can also be used to illustrate additional information. And I'm gonna show phylogenies that mostly have this shape on the right. So again, here's a phylogenetic tree showing that this individual A is most closely related to B. This tree on the right shows that same relationship. A is closest to B, or clusters together with B, but these branches here are much longer. It's a tree, so these are called branches. So the branches of this part of the tree are much longer than those down here. And especially for the kind of trees I'm going to show, what this is showing is genetic differences. So C and D are closely related, but also much more similar to each other genetically than A and B. A and B are maybe twice as genetically different as C and D. So the trees can show different things. They show the same relationship, but the one on the right also shows you how genetically distinct they are. And all the trees I'm going to show you today have that format where the length of the branches gives you information about how genetically different they are. Okay, so the way that we build these trees is we take the snail tissues that we collected from our surveys. So we sample them in the field, we bring them back to the lab and pick through them. And then we take those snails, they're preserved in ethanol. We take those snails and we do a DNA extraction. So we put the snail tissue into some salt and soapy extraction buffer, which will essentially break up the cell membranes and all of the other cellular structures and leave clean, pure DNA behind. We then do a technique called polymerase chain reaction. So this is also abbreviated as PCR. And what this is going to do is let us make lots of copies of just a couple of genes that we target. And so we're going to target one mitochondrial gene and one nuclear gene, and just make lots of copies of those 2 genes and then sequence them. And so we send our sample off for sequencing, and what we get back looks like this little diagram in the middle. So this is called a chromatogram, and each of these peaks corresponds to a different DNA base, an A, C, T, or G. And the order that they're in is the order of the DNA sequence of that snail for the gene that we were targeting. So we look through all of those and make sure they're of good quality, and then we line them all up. So this is a DNA sequence alignment. Each of these individuals, each of these rows is an individual snail, a little chunk of one of their genes, and we line them up so that each base pair is lined up with the matching base pair of all the other individuals we're comparing. And that gives us information. So for example, where I've got the arrow here, individuals 1 and 2, they both have a T at that position where everybody else has a C. And that's the sort of information that we're going to use to suggest that 1 and 2 may be more closely related to each other than to all the other individuals that are there. And we would reflect that. That's what would go into building something like an evolutionary tree here that shows 1 and 2 are closely related. And then of course we expand that out by several thousand base pairs in order to build out a big DNA tree to understand the relationships of these organisms. Okay, so now what I'm going to do is revisit each of our questions, tell you what our prediction is and what that should look like on a tree, and then show you the data that we found, what we have found so far. And just up front, for those of you who are thinking about this data, a little bit more familiar with this data, this is just CO1 data for these guys. So far, we're adding in the nuclear data, and we don't have complete sampling across all the cave snails yet. We still have several more taxa to include. So this is sort of preliminary. We've got another year's worth of data collection before we're ready to have a final story and publish it. This is what we think right now based on the data we have. Okay, so one of our questions was, are the cave snails all close relatives or have multiple lineages adapted to cave conditions? How many times have these snails invaded caves in central Texas? If they are all one lineage, we expect them all to cluster together on the tree. So they'd all be in one little colored portion of the tree. If not, if they are multiple lineages, they show up in different parts of the tree. So let's see what we found. Is there one invasion of cave conditions or multiple? All right, so on the tree, I'm going to show you surface snails are in black and cave snails are in orangey red. And so if you look at this tree, again, if they only invaded cave conditions once, they should all be together in one part of the tree. But what we see is there's a big clade down here, another one here, 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 and here. So looks like there are multiple lineages in these snails, this whole family of snails that have invaded and adapted to cave conditions. And then in some cases diversified, had more speciation happen after they got into the caves. So it looks like this group of snails has moved into cave conditions on multiple occasions. Okay, the next question that we wanted to look at is, how does shape evolve in these cave snails? The relatives of these snails all have tall shell shapes. So did the flat, like this guy, or the uncoiled shells, did they evolve once or did they evolve several times? Okay, so just so you know what to look for, from a tall root, this is an example of what the tree would look like if this flat shape only evolved once. Everybody in these other clades is tall, or these other clusters is tall. Then here you have flat shape evolved at this point, and then all the descendants have a flat shape or some derivative of that flat shape. That's what it would look like if flat shaped evolved once. If flat shapes evolve more than once, then they would pop up in different parts of this tree. So let's see what our cave snails in Texas seem to be doing. All right, so let me show you where flat body shapes are. Again, they're all from a tall ancestor. Here's a flat snail. Here's a flat snail. Here, here, and there. So there are at least 4 different groups that appear to have evolved a flat shell shape from a tall ancestor. Now, this is not finalized. Like I said, we still have a couple other species to put in here, including a couple more that are flat. So but it looks pretty clear that flat shell shape has evolved more than once in the Texas cave snails. And this is kind of an interesting thing because it sort of parallels what we see in other aquifer systems. In Brazil, in Central Europe, in Tennessee, there's flat snails in underground aquifers that look just like this guy here, Phaerodrobia micra. So this seems to be a common theme in cave snails. They get underground and then some of them diversify into this flat shape. Okay, and then the last question that I wanted to address with this phylogeny is the one where we were trying to get at initially. What species are there in the underground habitat and how do we tell them apart? So just so you know what our baseline hypothesis is, individuals that we have DNA sequence for that are representatives of the same species should cluster together. So for example, in this tree, all of these individuals shown here on these branches should be species 1, species 2, and species 3. So individuals of the same species should be closely related to each other. So that's what we expect. And that's how we tell if things are part of the same species or not. Okay, so I remember that text message from Pete asking me, what's this thing in Comal Springs? I really hoped it would be Stygopurgus bartonensis, the Barton Springs snail, because we really need more populations of that snail. What we found is Stygopurgus bartonensis is this little cluster down here. This is some from Barton Springs and another spring in the Austin area. So that's Stygopurgus bartonensis. And as you can see by the length of these branches, the members of those two different populations are not really very different from each other. Maybe just 1 or 2 base pairs of DNA sequence difference. So that's about how much difference you expect within a species. Now, our unknown from Comal Springs is clearly not the same as Stygopurgus as the Barton Springs snail, because it's in a totally separate group. If you look at it, the shape isn't quite right. Some of the sculpture is similar, but it's not the same thing. I was hopeful, but it looks like it's something, we can't say that it's the Barton Springs snail. It has to be described as something new. So now my question is, okay, so it's not Stygopurgus, but what is it? What genus is it? So if I put it in the context and add back in the names of all of the other relatives that are found on the surface in this family, We see some snails, this little group up here, that are from surface springs. So notice that this red little group in red, this is a cave snail. Everything else in these lineages that are colored black are surface snails. So they live just in surface waters like streams and lakes. So this clade up here, this little cluster up here, are from hot and salty springs in West Texas. This little group right here, that's the one that's closest to our unknown, are from estuaries on the east coast of the United States, like estuary Maryland. These guys down here are from Coahuila, from thermal springs in Coahuila. And these guys at the lowest part are also thermal and saline springs from New Mexico and Texas. So it's this unknown from Comal Springs. Oh, the other thing I wanted to point out is that these shells are scaled by size. So this guy is like a fifth of the size of all of its closest relatives. These surface taxa have eyes and pigment and all sorts of useful surface snail features. This guy from Comal Springs is depigmented and blind. It's a real cave snail, but it's most closely related to all of these other surface things. So what we have come to conclude is that this is not just a new species, but will have to be described as its own genus. So that thing from Comal Springs, one of the best sampled springs in Texas, is a whole new genus and species of spring snail. And we still have more to go. So let's sum up the answers to these questions as we wrap up and move on to the surface stuff. So some of the answers so far. Are all of the cave snails in Texas one lineage or several? And it appears clear that they've invaded cave conditions several times. Shape appears to evolve pretty easily towards a flat shape in these snails with apparently multiple evolutions of a flat shape. And then were these unknown things new species? And the answer to that is clearly yes, new species, new genus, and probably several of them. And so that's where we are right now. Several of our unknown things remain to be sorted out using this type of sequence data. And also the new Army Corps project is going to fund us to do some phylogenomic data analysis. We'll be working in the lab on describing this Comal Springs unknown as a new genus and species, and then figuring out what the heck all these other snails are that we still don't know what group they belong to. Okay. So that was some of our most recent discoveries about the cave snails in Central Texas. So now I want to move on and tell you about a project that we've been working on in surveying springs in West Texas and doing some DNA barcoding also to understand which species are present there and getting some more fine-grained data about their that's applicable for their conservation. Okay. So this part of the talk is going to be about snails that live in surface waters. So instead of being in underground aquifer and caves, these live on the surface in streams, and especially where there's spring waters coming up, these tend to really aggregate around springs. So these are in surface water. And this is with a group of people at Texas State University and U.S. Fish and Wildlife Service, along with some of my master's students and undergrads at UTRGV. Okay, so the goal of our spring survey work in West Texas is a little bit different from the Central Texas work. So in West Texas, there are many springs that have been very, very poorly sampled, and we know that most of them are going dry. You might have heard in the news about how the western part of the United States has had record drought. Things like Lake Mead are at very low levels. That's groundwater that people and also the organisms that live in that groundwater depend on. So in Texas, there's over 50 species of imperiled animals that are dependent on groundwater. Groundwater is also heavily used for people for things like municipal drinking water, irrigation, etc. So people use it too. And so there's potential for conflict, and the other potential for conflict is heavy use of groundwater by oil and gas in West Texas. And so all of those contribute to a decline of groundwater and potential decline of the organisms that depend on it. And so there's 50 species in Texas that have been identified as imperiled because they're dependent on these declining water resources. So the objectives of this project were to survey about 200 springs for that have been previously unsampled for small and large invertebrate animals, and really emphasize the snails. We're emphasizing the snails, obviously that's my interest, but also because there are more snails than any of the other groups that are already listed by the U.S. Fish and Wildlife Service as federally endangered or threatened. And so the snails are a special interest for state and federal agencies because some of them are already listed. So we're emphasizing the snails to try and build out our understanding of their relationships and what species are present. Okay, so we're going to survey a bunch of places. Then up front, we're going to develop a DNA barcoding library. So essentially sequence the DNA of all the snails that we find, use that to build an evolutionary tree, and that will let us know which species are present where and allow us to identify any new snails that are found. Then the last step of this project will be to provide provide Texas Parks and Wildlife with a ranking and priority of the species that we encountered, and also prioritize the springs in terms of which have the most highly imperiled organisms. So that way, out of 200 springs, they have limited conservation dollars. They can decide which are their top 5 or 10 areas that they really need to focus their effort on, which are the most highly imperiled. And so that's the bigger picture of this project. The snails that we're focused on are called spring snails, again, surface snails, not cave ones this time. They're all freshwater. And some of them look like this. This is Pergolopsis, also called pebble snails. And they're smaller than pebbles. These are 3 grown-up adult individuals on the end of someone's finger. So you can see how small they are. They're not cave snail small. You can see these in the field, but they're still pretty small. You can get 10,000 per meter square. They can reach really high densities in their preferred habitats. Okay, so to start off this project, what we did was get on Google Earth and map out the known springs in West Texas based on the book Gunnar Brune, which is The Springs of Texas. And then we also spent a lot of time zooming around on Google Earth and looking for other places where springs were likely to be found. So what are land formations that look like springs should be there? And then is the appropriate vegetation there where it looks like, out in this parched desert habitat or landscape, if you see a little cluster of trees, that probably means there's water there part of the time. So we went and scanned around on Google Earth looking for springs, mapping all those sites. I'm going to show you a picture in a little bit from the bottom of this canyon. So you can see this is what it looks like from the top. We said, all right, maybe there's some water in there. We can't really see because there's a shadow. If you go backward and forward in time on Google Earth, you can see sometimes there's water in there. So we put it on our map and said, okay, we're going to try to hike to that site and see what we find. So we map all those sites out. This is again on Google Earth. And then, all right, we have this 200 sites identified. We need to figure out how we can get to them. And first off, we need to figure out, can we sample them? And so we put an overlay on our GIS project of who owns the land. Most of the West Texas is private land. And so we had to kind of focus our sampling in areas that are state parks or federal parks, or that have landowners that will allow us access. And so some great looking spots we may never be able to get to because a private landowner is not amenable to us coming and sampling at their springs. So anyway, we identify our sites, we figure out who owns it, and then the next thing we're going to do is pack up our gear and take a long walk. So we drive the closest we can get to the field site, unload. It takes at least 3 people to carry all the gear, and take a long walk to wherever we find water. And so this picture on the right, this is one of the Texas State students, Lucas, that did the work with us, has been doing the work with us. And this is showing you the canyon from the canyon view of that Google Earth image I showed you before. So from the top, we couldn't tell if there was water. What we found out sampling there is at the very top under this cliff ledge, there's a pool of water that comes out from underground. And then it kind of trickles down this canyon down to where it dries up in the arroyo. So we were able to take our samples here and climb all around all of these boulders up to the top of the canyon and sampling for animals and invertebrates as we went. Now, the type of samples that we take here, we're on the surface, so we don't have to dig wells or anything. So we're doing a little bit different sampling techniques. One of the qualitative sampling methods that's just going to give us species richness, look at overall diversity. We take with what's called a dip net. So you take a net and you kind of bang it around in different parts of the stream, trying to get the biggest picture of diversity that you can. We're also trying to compare measurements of water quality and aspects of the water to help us understand what drives patterns of species richness in these springs. And so at each of the sites, we take water quality measurements, things like how much dissolved oxygen, how saline the water is, how cloudy it is. So we take all those sorts of water chemistry measurements in the field. And then we also have some quantitative estimates. So these are what will allow us to compare not just species richness, but also relative abundances across sites. And so on the left, you see a little image of a sampler called a Cerber sampler. It's a standard aquatic ecology sampler. So you have a net that's attached to a little square of a known area, and you kind of agitate all of the rocks and leaves and fluid here in the square and catch everything that floats by in the net. And then you've sampled a known surface area and can compare across sites how many animals you find and what kinds. And then we also did our hyperrheic Bow-Rausch sampling, where we pound a spike into the gravel and then pump the water out. And sometimes that runs into problems. So for example, here we'd hiked out 2 miles to a site in Big Bend Ranch State Park and then broke the head off our sledgehammer. And so we'd only taken 1 sample at that point. So then we spent the next 3 hours pounding in the spike by hand, or Lucas did most of it. So sometimes the sampling is pretty arduous. Now, the snails, the spring snails that we're interested in doing DNA work for and describing new species, we collect those by hand and then process them separately from the rest of the samples. Essentially, all of them are preserved in ethanol until we're ready to work with them back at the lab. Now, the field samples, if I take a dip net sample in a high quality habitat, that dip net may have 10,000 tiny animals in it. And an underestimate would be 10,000 tiny animals. And that could take a month of sorting through and picking and identifying. So we cut that down a little bit by hunting for the big rare animals and then cutting that sample into a quarter and doing what's called a subsample, where we're just going to pick through and take all the animals out of 1/4 of that sample and then extrapolate that to understand the entire sample. Okay, so where we are so far on the survey broadly, we've surveyed 65 springs, 222 samples, and we're about 1/3 done with processing and ID. So I'd say in a good year and a half, we'll know exactly what we found. But so far we've focused up front on the snail stuff. So that's the results that I want to tell you about so far. All right, so there's 2 genera of spring snails in West Texas, and several of these species, as I'd mentioned before, are federally listed. So they're considered threatened or endangered. So I wanted to tell you a short story about Trionia. Trionia are sort of the elephants of the snail fauna. They're ones that tend to go extinct. And so, for example, we have 4 species in Texas. Trionia oasiensis is already extinct, and so is one from Phantom Lake. We have a couple other species. The one I wanted to focus on is Trionia cheetami. This is the phantom Trionia. So maybe some of y'all have heard of Balmorhea State Park, which is a big spring-fed pool in West Texas. And so this species occurs there. And it's in high numbers, like 10 million individuals, but mostly in this big pool that's very heavily used by people. And it's federally listed species. It has a couple of populations, one of which is in Phantom Cave. Now, Trionias, as I was saying, are elephants because they tend to go extinct. They give birth to live young. So an individual female may only have just a handful of offspring over her life. They are also directly compete with another invasive snail for food. And they like the edges of springs, like the silty parts at the edge of a spring, which is the part that people tend to go and channelize and put concrete on their favorite parts of the spring. And so these are more likely than other snails to go extinct. Okay, so if we, as I said, we're sampling for these to build out a DNA barcoding tree. And so here is the tree just based on the mitochondrial gene. And so this is just showing Trionia in Texas. So here's one species of Trionia. Here's another one, the one that's gone extinct, and then some of distant relatives. Now, Trionia cheetami, this is the one at Balmorhea State Park. As I mentioned, they have a couple of populations. One in Phantom Cave, and Phantom Cave population has actually already dried up. it's currently maintained by a pump where they pull water from deep in the cave and feed it out to a pool outside, because that spring supports like 5 federally listed species, including a fish and a crustacean and a couple of snails. So this Phantom Cave spot has actually already sort of artificially maintained. And then there's the ones at Balmorhea State Park. So that's this little group right here. And you can see these are pretty shallow nodes. So these are pretty closely related. There's about 1.2% sequence differences between these two populations. And in this group, that's right at the edge of what we consider to be the usual differences among the same species. Since these are federally listed and people were really getting worried about, especially the Phantom Cave population, because if that pump fails, that species, this population dies out, they wanted to know, can we take individuals from the other population and use them to repopulate Phantom Cave if we need to? And so we did a little bit finer grain analysis to try and understand the amounts of gene flow among members of these two populations. Okay, so again, here is, I'm just focused in on that part of the tree, the ones up here from Phantom Cave and below from Balmorhea. Now, some other work that one of my current grad students, Christina, had worked on quite a bit is trying to understand the reproductive biology of these two populations. And she actually found that in addition to being slightly genetically distinct, they also have slightly different breeding seasons. So they do have some real differences among them. So what we want to know is, are they useful substitutes for each other? And so what we did was do a technique that lets us look a little bit more carefully at how much gene flow there is among populations. And this is a technique called genotyping by sequencing. Essentially, we're going to take a similar process where we get the DNA from many individual snails. We're going to extract the DNA. And then instead of targeting 1 or 2 genes, we're going to just chop up that DNA into millions of pieces, add little tags to those pieces, and sequence them all at once, and then use bioinformatics methods to reassemble the genome and understand the DNA sequences. So this is a different method called genotyping by sequencing. Instead of just having a couple of thousand DNA base pairs to compare, this results in a couple of million DNA sequence base pairs to look at. So it gives us a whole lot more information, but it's also pretty analytically challenging. The bioinformatics part of it is challenging to work with. Okay, so on the graph I'm going to show you, this is not another tree. This is actually a principal component analysis plot. So on this graph, what you're going to see is each little dot represents a snail, the genomic data from a snail, and there are going to be some from Phantom Cave and some from these other populations, including the ones at Balmorhea called San Solomon Springs. On this type of plot, a PCA, you'll have essentially individuals that are more genetically similar will be dots closer together. If they're less genetically similar, the dots will be further apart. So closer is similar and further apart is different. Okay, so the Phantom Cave dots, those ones from this little clade up here, are these purple individuals. And as you can see, they form a pretty tight cluster. There's one over here that's getting close to the other group. This group in pink is all of the other populations of Trionia chaetae, the Phantom Trionia. And as we can see, they kind of separate out by the population they're part of, but they're also intermixed. And so what it looks like is all of the snails from this lower group are probably one management unit. All the ones from Phantom Cave are another one. So what this suggests to us in terms of conservation is that these probably are not suitable substitutes for each other. And whether or not they're different species, it's kind of on the fence, but they definitely should be treated differently. It's probably not a good idea to take individuals from Balmorhea, raise them in an aquarium, and put them into Phantom Cave. That's probably not going to be in the best interest of the species. So whether or not they're different species, they probably are different management units. There is restricted gene flow between those two big populations. Okay, so that's Trionia's story, and one where we've gotten into a little bit more depth. I wanted to wrap up by telling you about one last story of species discovery, some of the pebble snails in the genus Pergolopsis that we found in this region. Now, Pergolopsis are snails that are super diverse. This is the most diverse group of freshwater snails in North America. There's over 200 species, most of them in the American West. When we map out some of the species that are found nearby Texas and the Big Bend area, there's a lot in New Mexico and Arizona. There's quite a few in Mexico, but there's this big gap here in the Big Bend region of Texas. And so one of the things that we wanted to know is, are there no Pergolopsis in that region? Or has no one just gone and looked for Pergolopsis in that region? And so we've kind of concentrated some of our sampling looking for members of this snail group in the Big Bend region in Texas. Okay, so what we found so far is 5 new populations of Pergolopsis or pebble snail. These are the ones that are known, these little blue dots, they were already known. And then we've just recently described 2 new species of Pergolopsis. I'm going to show you a little bit of the data that we use to describe them. So this is Pergolopsis hairy milleri from the southern Sierra Vieja mountains, and then Pergolopsis rubra from Palo Amarillo Springs in Big Bend Ranch State Park. And then just a few months ago, our trip in October, we found 2 new populations that we're not sure if they represent new populations of rubra or new species. So we're still in the work of doing the work of characterizing these. And I also want to mention we're not quite done. We have a trip coming up in April. If you see this little gap right here along the Rio Grande, this is real country that has real canyons. And so you can't get down to the river except by floating up from upstream. And so we're going to spend 2 weeks floating down the canyons of the Rio Grande and sampling all the springs that run into the river. So we're hoping we'll find additional populations here in the next month or so. But anyway, so let me tell you how we decided that these 2 populations, this triangle hairy milleri and this square rubra, were new species, what evidence we used. Okay, so now you know how to read phylogenetic trees. This is showing you on the left, the mitochondrial tree, and on the right, the nuclear tree. So this little brown cluster is Pergolopsis metcalfi, one of the known species of Pergolopsis. This cluster in pink is Pergolopsis rubra individuals, and this one at the bottom is our other potential new species from Vasquez Springs, from the Sierra Viejas. And so what we found is there was what's called congruence among our genetic data. So both the mitochondria and the nuclear data told us the same story, that these appear to be distinct. Each species is most closely related to other members of what we think is that species, like I showed you before on those hypotheses. Now, some of the other data that we use to describe them, and what we'll use to describe all of the new species I've told you about today, is we also looked at the shapes of the shells. These are, again, graphs that are similar to a principal component analysis, like I showed you before, where dots that are very close together represent individual snails. In this graph, though, the data that underlies it is the shape of their shell. So instead of the genetic or genomic data, this shows you that these 2 individuals of Pergolopsis rubra are very, very similar in the overall shape of their shell. And what we found is that all the individuals of our proposed new species, Pergolopsis rubra, are very similar in shell shape. Hairy milleri and the known species metcalfi, we can tell them apart by their shells. We also use a couple other anatomical features to distinguish them. So for example, this is the shells, and then here it shows you the snail's teeth. This is the structure called the radula, or the scraping structure that they use to graze. And here's a little tooth, the cusp of a tooth of the known species. Metcalfi is kind of curved. On our new species, rubra, it's really straight and pointy with a wide base. A lot of some of the most quickly evolving characters in snail anatomy, and ones that a lot of the naming is based on, is male and female genitalia anatomy. And so one of my master students is a really amazing scientific illustrator. And so he did these scientific drawings of the structures of the male penis, which include several lobes, and then these little structures marked with stripes that are glands that secrete different secretions. And so in metcalfi, they're known for having a terminal gland on this lobe, but in our new species rubra, that gland is not at the tip, it's not terminal, and it wraps around the back. They have a unique orientation of their pineal glands. Then finally, just the color. This metcalfi, the known species, is kind of brownish gray, and our new species, Pergolopsis rubra, is red. So we named it rubra for the red rocks and red pigmented color of that species, the red rocks of the canyon where we found it. Okay, so this is that canyon. This is Palo Amarillo Springs and Big Bend Ranch State Park. And just so you have an idea of how big these spring systems are, Here is my student Houston, who did the drawings, sitting at the stream side. There's a little pool over to his right, but this is the width of that stream. It's basically one cattail wide. And then this is the waterfall where this snail occurs. The picture on the left is looking up that waterfall. So it's a waterfall that's maybe 5 feet across and has a little trickle of water. Right here, the base of the cattails, and in all of this vegetation in the water trickling off this canyon wall is the entire range of this new species. We looked up and down this canyon. It's not anywhere else. As far as we know, this is the only place on Earth that it occurs. And then our other new species is actually even more ridiculous of a range. So these guys, this is some of my collaborators, are standing around the whole range of the species. The water emerges from this boulder on the right, and then it's a couple of millimeters deep and maybe a foot or two wide, and trickles down until it dries up right here. So their entire range of the species on this really desolate and dry mountainside in the Sierra Viejas is this is their whole habitat. And while we were at this field site with the landowner, we could tell that the site had been trampled by livestock, especially by aoudad, which are an exotic game animal that's been released in West Texas that people hunt. And that sort of trampling by livestock is really known to decrease snail populations because it kind of messes up all of their food source and they get smushed and it messes up their habitat. One of the nice things about working with this particular landowner, Harry Miller, is that he has agreed, once we describe the species, to fence off the upper portion of this stream to really protect that land better from some of the cattle grazing and trampling. Okay, so where we are now with that project. As I told you, we wanted to sample a bunch of never sampled springs for small and big invertebrates. We're about 3/4 of the way done. We wanted to develop this DNA toolkit to be able to understand relationships among the known spring snails and identify anything new that we found. That's pretty much done. As we find any new populations, we add them in. And then finally, we want to rank and prioritize the species in the springs and tell Texas Parks and Wildlife what's their top conservation priority. That has to wait until all the data's in. And so we have to stop visiting springs and collecting new data to get that answer, but that'll happen in about a year. Okay, so I just wanted to wrap up before we get to any of y'all's questions and discussion by acknowledging all the folks who have funded and worked on this part of the project. Mostly the money's from Texas Parks and Wildlife. And then UTRGV greatly supports the project. We've had help from lots of landowners, people who came out and helped us collect samples and helped us get access to land. I also want to thank some of the folks in my lab. I showed you the names of some of my current students earlier. Here's also some pictures of past and current students out sampling. So here's the lab group saying goodbye. So now I'd like to take any questions that y'all might have. I'll let this little video of Pergolopsis run while we chat.
Thank you so much for this interesting talk. We kind of noticed, I think, even with chatting with some faculty, we did not know, I think, anything about snails because this seminar was very informative. We had some interesting questions. However, a lot of them you actually did an awesome job answering throughout your talk. But I do want to start with this one. This question from Tori. Do you have a favorite species of snail?
You know, I really like this guy here. So this snail here in the middle of the slide is Phreatodrobia spica, the spiky cave snail. This is one that we described from Comal Springs, and I wrote up that description with my former student Dominique Alviar, and she was an undergrad who worked with me, and then is now at Texas A&M working on her graduate degree. But it was just a really fun one. My friend Pete said, "Hey, do you know what this is?" And I said, "No, I have no idea." Clearly nobody knows what this is. And so we all worked together for a couple of years to put the data together to describe it, and had a lot of collaborations, and it was sort of a real capstone project for that undergraduate student to complete to describe that species. So that's probably my current favorite.
Awesome. I like the question because you guys are exposing to a lot of different types of snail. Something kind of similar, but I found it interesting from Wendy Rodriguez is you mentioned about your favorite sampling method, and she's asking if that method is more effective than the others.
Well, each of them are going to sample different habitats, and perhaps, not all of the snails are going to be in a cave. Some of them are just going to be in habitats that are like layers of the aquifer that are way deeper than the caves go. And so, you actually have to use a variety of sampling methods to sample all the different habitats, and they're going to have their pros and cons.
Okay.
That was my favorite because I can take the family and we can all have fun swimming in the spring at the end.
Okay. Interesting to know that. So the next question, actually a few students were asking relatively the same thing. And a student actually shared that they learned that snails contribute to the ecosystem in a variety of ways, and one of them being the source of calcium for other species.
Mm-hmm.
And that student and other students are actually asking that what would be the impact to the ecosystem if these snails were to become extinct?
Mm-hmm. Yeah, so as you said, they contribute calcium. So they put some of the calcium from the water and the rock around them into their shells. So they contribute calcium to other organisms. But they probably more importantly contribute their carbon to other organisms. So you can think of them, if you think of a real simple food web, there's the bacteria that are the primary producers, the base of that food web. And then there's things like the snails that are grazing on bacteria. And then they're going to be eaten by salamanders, fish, and shrimps. And then those things are going to be eaten by bigger fish. And so they're part of that entire ecosystem. So if the snails are no longer there, then you don't have any way to get that nutrients from all of the bacteria, the primary producer, to the higher levels of the food web. So there's that potential breakdown of their little ecological network if they went extinct. And so one of the reasons people focus on snails to get information about ecosystem health is they kind of consider them a marker, ecosystem marker, or like a canary in a coal mine sort of for ecosystem health, because they also have pretty narrow tolerances to pollution or things like changing dissolved oxygen or water quality variables. So if water quality changes drastically, all of a sudden you don't see snails anymore, you have a potential long-term water quality problem.
That's interesting to know how important they are, because some students were actually also questioning about why research snails. And what was their importance. So that's very interesting. The other set of questions that I'm putting together is about specifically the Rio Grande Valley. Mm-hmm. And one of the questions is simple. What is the most abundant known species in the valley?
So probably that is an introduced species, Melanoides tuberculata. So if you ever see where they've gone and dredged out, the city has gone and dredged out a canal and like dumped the canal spoils on the side of the canal, you'll see these tall, maybe they're an inch tall and very thin snails. Like they have a shape like this. Those are probably the most common, and that's an introduced species. The most common native is one called Physa, and they are one, if you pick them up, they actually have their opening to the left. They're one of the few left-handed snails, and that's kind of a common pond snail that's found worldwide. So those are probably the most common freshwater snails in the valley. The most common land snail in the valley, it's found in every one of your yards, and you can get 10,000 of them like under every footprint on your lawn. And it's a little tiny snail called the snaggletooth snail, the Gastrocopta procera.
Interesting. So this comes to my next question. would you say that the most common was an invasive species? If not, then which, especially from the ones that you've been discussing, do they have an invasive species?
Yeah, the most common in the Rio Grande Valley would be that invasive species, Melanoides tuberculata. And that is one that's originally from Asia and has been introduced worldwide, and it lives in freshwaters throughout Texas. It's one that directly competes with some of those West Texas spring snails for resources because they eat the same thing. So that's the most common, one of the invasives. And
yes,
yeah, go ahead.
No, go ahead. I'm sorry.
No, I was just going to say, it does appear like that they directly compete with some of the native snails for resources. There's not any evidence, though, that that interaction has ever driven any of the native species extinct.
Hey, that's good to know. My other set of questions is about, everyone was kind of concerned, I guess, mostly because you were sharing how the process of your research is going, not only sampling, but if so, from the Rio Grande Valley, are there no species of concern?
Yeah, so that's a good question. In terms of freshwater, there are freshwater mussels, clams, that are species of concern. There are not freshwater snails that are species of concern. The real species snails of conservation concern in the Rio Grande Valley are terrestrial species. So we did a big multi-year project surveying and understanding describing the diversity of land snails, terrestrial snails in the Rio Grande Valley. And there are quite a few that are of conservation concern, and that we found lots of neat places in the valley that really have good examples of those. So for example, the Ceiba Palm Sanctuary in Brownsville is the only place where one species of snail, the Rio Grande snaggletooth, has ever been found alive. And then there's another species called Hubbard's land snail that's the only place in the United States it still lives. It's one that needs intact sabal palm groves to survive, and those are the only ones left in the United States, and so that's the only place that snail lives. And then there are, maybe you guys are familiar with a snail that's about 2 inches long and brownish red that you sometimes see crawling around on the sidewalk in the city. That's a carnivorous snail called Euglandina texana, which is called a wolf snail. They're carnivores and they only eat other snails. That's one that the state has kind of wondered if it was becoming threatened. Often carnivores get threatened more quickly than lower trophic levels. And so that carnivorous snail is one that they were really concerned about its conservation status, that South Texas wolf snail.
Okay, interesting. And if there are, and like I said, a few students actually were asking about this, if there are some that are potentially endangered, these methods of extracting them, wouldn't it be harmful to that ecosystem?
Yeah, that's a good question. So, and it's going to be different whether we're talking about the cave environment versus the surface springs. So in the cave environment, the samples that we're taking are mostly we're mostly taking samples at springs or wells where the snails that are coming out that we're collecting are the ones that were getting kicked out of their home already. They're being ejected from the aquifer. So they were about to die before we caught them. So in that case, we're sampling stuff that was on its way out already, getting kicked out of the aquifer. So there's no real concern there about whether or not we're taking viable individuals from the population. And in the spring situation, though, it's a little bit different. Now, in good spring habitat, you can get 10,000 tiny snails per meter square. They can reach huge densities. So if I take 10 or 20 for my research, it does not have a huge population impact. And the fact is that that spring cannot be conserved unless you have some legal regulatory reason to conserve it. Like you can't tell oil and gas company, sorry, you can't pump that spring dry because we don't want you to. You have to have the regulatory reason. There has to be some federally listed threatened or endangered species there, or you don't have any reason to tell them not to pump enough water that the spring goes dry. So if I kill those 20, it's has the potential to save the species. If there's a new species there, we describe it, and then there's a reason that the state or federal agency needs to say, you need to mitigate your water usage here and pump the water, but don't dry it completely. So I guess, yes, killing 20 of them is an impact, but usually small and for the greater good is what I would argue.
Okay. Yes, I found it interesting that a lot of students or a lot of our members were mentioning this and kept writing it.
We're concerned.
They're concerned.
Mm-hmm. Yeah, I understand. And we don't take more than we really need to. And especially in places where they're very rare, you collect and you only take every 10th or every 100th that you run across to make sure you're not taking too many from a population. So it is something we definitely think about.
That's good to know. And my last question, I know that it's for benefit for all of our students, but Paula Guerrero actually asked it. How can any of our students start to and participate in any of these research?
Sure. So, we actually had been talking with your department chair a little bit about a research exchange where current STC students could come work in labs at UTRGV in the summer. And so I would talk to Dr. Cervantes about that as a first step for current STC students. And if you are going to be coming to UTRGV, what I advise the students to do is to look at the biology department website and see what sort of research all the different faculty do, and then read a little bit about what they study, and then email them and say, hey, I'm looking for, politely in a nice formal email, I'm looking for a research experience. I'm very interested in your work. Could we set up an appointment to meet? And sometimes that professor might have a full lab and they can't help you that semester, but often people are looking for extra hands to contribute to work. And students take credit hours, so a research course, like a regular class, a 3-credit research course to get experience and some credit for their time. Sometimes they work in paid positions, and we would send emails out to the bio majors emails that tells them anytime someone has a paid position open, we'd say, hey, we're looking for students, please send me an email. And so I would watch out for that. Once they were at UTRGV, look at the faculty's websites and say, hey, I'm interested in doing some research, and I have some time to give and I'd like to learn.
Well, thank you so much for agreeing to joining and speaking about your research in our speaker series. Mm-hmm. I want to tell everyone, thank you for joining us to actually subscribe to our channel, the Biology Department Speaker Series. We will have a seminar next week, so we will be sending out those flyers soon. Thank you so much, Dr. Perez, and thank you everyone for joining us.